Genome annotation consists of two steps. One is structural annotation to identify gene regions, and the other is functional annotation to assign functions to genes. As you know, protein coding sequences start with a start codon ATZ and end with one of the stop codons. But there are many candidates that meet this criteria. It is necessary to distinguish real genes from fake genes. The two major strategies for gene prediction are ab initio method and evidence-based method. The former one is based on the gene signal and gene content, and the latter one uses expression data or homology to known genes. Since the gene structure of prokaryote is relatively simple when compared to that of eukaryote, ab initio method works well for prokaryote genomes. This shows an example of GenBank format file, one of the standard formats to describe annotated genomes. This file includes metadata part describing information about submitter and reference. Annotated genes are described as a feature. CDS feature contains information about protein coding genes, which includes location in the genome sequence, amino acid sequence, cross-reference to related databases, and so on. For prokaryotic genome annotation, many automated tools are available. Among them, we will use DFAST web server in this lecture. DFAST uses default standard gene prediction tools shown here for structural annotation by employing ultra-fast homology search tool GhostX. DFAST can rapidly conduct functional annotation. The total runtime of DFAST is about 3 to 5 minutes for typical sized bacterial genomes. Let's try using DFAST. First of all, download the assembly result from the supercomputer. And open the website. All you have to do is to upload a genome sequence file here and press Run button. Optionally, you can provide job title and email address. DFAST will let you know when the job is finished by email. To perform taxonomy and quality check, enable these options. The job has been completed in 4 minutes. Let's see the result. In this page, statistics of the annotation is shown. You can download files from here. In this page, you can browse all the annotated genes here. In the next page, you can create a date submission file to DDBJ following the guideline shown here. Finally, the result of taxonomy and completeness check is here. The organism name identified by DFAST is Lactobacillus hokkaidoensis. The completeness value is about 99%, and the contamination value is 2.5%, suggesting that the quality of this assembly is quite good. Now, we have finished genome assembly and annotation. Usually, comparative analysis or phylogenetic analysis will follow this using several annotated genome data. Recently, all-in-one tools are emerging. They can not only conduct assembly and annotation, but also conduct such a downstream analysis. Please try them if you are interested.